Measuring evolution in a population. Fitness increases favorable traits. Variation in population gives the population different traits that allow for variation in population can come about two ways within a population. One is mutation, where we have a random change in the DNA, and the other is through sexual reproduction, because we have a recombination of genes. If you remember back to biology with meiosis, you have the crossing over, which is the picture down here, where the homologous chromosomes are going to switch pieces and create that variation. And this is completely random of how much and what pieces of DNA are switched. Just some reminders of different terms that you'll need to know for this chapter, some that we have not gone over yet, but hopefully that you remember them from biology. What are the things that drive evolution? There are 5 things. We have mutation. Mutation creates variation in the DNA. Then we have movement, so movement of genes from one area to another, so migration, immigration, and emigration. We have natural selection. Certain allele populations increase fitness. We have non-random mating, sexual selection, basically looking for the best mate. And then we have small populations. When we have a small population, there's an increased loss or an increased gain of particular traits because of genetic drift. There's a way that we can actually measure our population change using Hardy-Weinberg. There are 5 things for Hardy-Weinberg equilibrium. We are working on a non-evolving population, so we have no natural selection, we have a large population, we have no movement of alleles, we have no mutations, and then we have random mating. For Hardy-Weinberg, we have 2 equations that we need to know. We have P plus Q equals 1, and we have P squared plus 2PQ plus Q squared equals 1. In this equation, we are looking at the frequency of alleles in a population. P is the frequency of the dominant allele, Q is the frequency of the recessive allele. We're going to use big A as our dominant allele and little a as our recessive allele. P squared is going to be the frequency of our homozygous dominant, 2PQ is going to be our frequency of our heterozygous, and Q squared is going to be the frequency of our homozygous recessive. When doing Hardy-Weinberg problems, basically all you need to do is plug in what you know and then solve for the unknown. Let's go ahead and go through a problem real quick. Let's say we have a population of flowers. We have a red gene and a white gene, and it is incomplete dominance where the red and the white gene together will cause pink. We can take a look at the frequency of alleles. 80% of the gene pool has a red gene, 20% of the gene pool has a white gene. So that means our P is going to be 0.8 and our Q is going to be 0.2. We then could figure out the frequency of each combination. We have our P squared, that's going to be RR, that would be 0.8 times 0.8, giving us 0.64. This would be 64% of the population. We then can find our Q squared, which is going to be our WW, that would be 0.2 times 0.2, giving us 0.04. This is 4% of the population. And then we can find our heterozygous, which is going to be PQ, that's going to be RW, that's going to be 0.8 times 0.2, which is going to give us 0.16. So this is 16% of the time, and this is 16% of the time. Since it's 2 of them, it's going to be 2PQ. The possibility of being heterozygous for this trait is going to be 0.16 plus 0.16, giving us 0.32. 32% is going to be the heterozygous in this case. These are the things that you should know.